Hello, and a very warm welcome to everyone joining us for today's Select Science webinar entitled Quantitative MicroRNA Expression Analysis in Tissue and Body Fluids, Whole Genome Profiling using Small RNA Sequencing followed by qPCR-based validation. My name is Sarah Thomas, and I will be moderating today's presentation. I'm delighted to introduce today's speaker, Dr. Ariane De Ganck. Ariane is the Product Manager of Transcriptome Services at Biogazelle. Today she will be talking about the combined approach of whole genome microRNA profiling using small RNA sequencing and targeted qPCR-based microRNA validation, and will present case studies for microRNA expression analysis in challenging samples. Before we begin the presentation, I would like to ask you 4 poll questions about your research and the technologies you use. Please select the most appropriate answer or answers from the choices presented. Okay, so our first poll question: what technology are you currently using to quantify microRNA? And your options are QIAGEN, Hexacon, Life Technologies, I'm quantifying from my NGS data, or other. Okay, we'll move on to poll question 2. Where do you typically send your microRNA profiling work? Your options are: we perform inside our lab, send to a local or affiliated core facility, send to a commercial provider, we currently have not done any microRNA work but are interested to, or we currently have not done any microRNA work and have no immediate plans. Now we'll move on to poll question 3. Where in the research process are your current project or projects? Option 1, biomarker discovery, pathway or targeted validation, individual assay testing, functional validation of verified microRNA, or bioinformatics analysis of microRNA function. And we'll move on to the final poll question: what product strength is most important in your microRNA studies? And your options are dynamic range, sensitivity, specificity, cost, or other. Okay, thank you for your input, and we will discuss the results of these poll questions at the end of the webinar. Before I hand over to Ariane, I would like to mention a couple of things. Please feel free to ask any questions for the Q&A session at any time during the webinar by clicking on the Ask a Question button at the bottom of your screen. You can view the presentation in full screen by clicking on the tab at the bottom right of the Media Player window, and you can download related white papers in the Resources tab at the bottom of your screen. The on-demand link for the webinar will also be provided afterwards. Without further delay, I would like to hand over to Ariane, and I would like to thank her for presenting to us today. Please go ahead, Ariane.
Thank you, Sarah, for the kind introduction and the poll questions. Also from my side, good morning or good evening to you all, and thank you for joining us today. First of all, I would like to thank Select Sciences and QIAGEN for organizing this webinar and inviting Biogazelle to present today. During the next 45 minutes, I will discuss the workflows that Biogazelle applies for quantitative microRNA expression analysis in tissues and body fluids. Just a brief overview of the topics that will be handled. I will start with an introduction to non-coding RNAs and their potential as biomarkers, followed by a brief overview of the platforms that exist for microRNA expression analysis. Afterwards, I will focus on the workflows that we apply specifically for whole genomic microRNA profiling and targeted microRNA validation. I will finish my presentation with some information about the transcriptome services offered by Biogazelle. Biogazelle is committed to help its customers to understand the transcriptome. We believe that a better understanding of the coding and the non-coding RNA targets will help the identification of new biomarkers and therapeutic targets, and that this will help in turn to improve healthcare by next-generation diagnostics and therapeutics. So the company really focuses on the RNA level because we really believe in the power of RNA as biomarker and as therapeutic target. Why is this? So RNA expression reflects the state of a biological system. DNA analysis provides a static view of the biological status, while RNA information gives a view of the dynamic changes in cells, tissues, or body fluids. So let's say that DNA is the picture and RNA is the movie. RNA expression levels are highly dynamic and integrate both genetic and epigenetic mechanisms of gene regulation. To give an example, when treating cells or patients with a therapeutic compound, in many cases the DNA remains unaltered while the RNA will reflect the response to the treatment. Equally important, the technologies are available to measure the RNA in so-called gene expression studies. These technologies allow high-throughput screening using low amount of input material, which is, for instance, not the case for protein measurement technologies. This figure summarizes the transcriptome service portfolio of Biogazelle. Biogazelle offers a total solution for the study of RNA expression levels, starting from RNA extraction over measurement using state-of-the-art and validated technologies to understandable results. We offer these services for the most interesting RNA molecules, being messenger RNA, microRNA, and other small RNAs, and long non-coding RNA. The type of studies that can be discovery studies, in which we perform whole genome-wide screenings to discover targets of interest, but this can also be targeted validation studies for a limited number of targets of interest. Depending on the type of study, different technologies can be applied, including RNA sequencing, but also qPCR or digital PCR for the validation part. We do not only focus on the wet lab activities, but also offer an extensive portfolio of dry lab solutions to process and interpret the obtained results. As I mentioned before, we do not only focus on the protein-coding messenger RNAs, but are also interested in the non-coding RNAs. Let me go into these non-coding RNAs a little bit more. So we are all familiar with the cascade from DNA over transcription to translation. However, today it is clear that a large part of the genome are transcribed to RNA molecules and that are not translated in proteins. These so-called non-coding RNA molecules are involved in the regulation of the expression of proteins and protein-coding genes. Interesting to know is that there are many more non-coding RNAs identified today than that there are protein-coding genes. To give an idea, the type of non-coding RNAs that are best studied are the long non-coding RNAs or link RNAs and the microRNAs. And currently we have 2,500 microRNAs identified and more than 60,000 long non-coding RNAs, while there are only 21,000 protein coding genes. Moreover, the non-coding part of the genome of the transcriptome explains organism complexity. So what is shown on this graph is the correlation between the number of coding base pairs in red and non-coding base pairs in blue, and the complexity of the organism in terms of distinct cell types on the one hand and the genome size on the other hand. As you can see, while the genome size and the number of different cell types is increasing, the amount of coding base pairs reaches a plateau phase. while the non-coding part keeps increasing with increasing number of cell types and genome size. To give an example, the worm C. elegans with 29 different cell types has 19,000 protein coding genes, while a human with 169 different cell types has 21,000 protein coding genes. The difference between worm and man is largely explained by the non-coding part of the RNA. Another interesting characteristic to study non-coding RNAs is their potential as biomarkers. Non-coding RNAs have a highly specific tissue expression profile, which could definitely be of interest when studying this type of genes in cancer research, but also in other diseases. To give an example from the oncology field, it has been shown that it is possible to detect cancer-specific non-coding RNAs that function as lineage survival oncogenes. This means that they are specific, expressed in certain cell lineages and overexpressed in tumor cells derived from that lineage. These non-coding RNAs have a survival function in those cancer cells, meaning that if you knock it down, cells die. This is very interesting for two reasons. These are excellent therapeutic targets, because if you knock them down, the cell will die, and because of their specific expression, you don't have to worry about unwanted side effects. But they are also excellent diagnostic or predictive biomarkers due to these cell-specific expressions. In addition to this, these non-coding RNAs are also present in the circulation, where they are bound to proteins or packed in vesicles that makes them stable in circulation. It has been shown that the expression levels in circulation predict disease and correlate to patient prognosis, which makes them excellent, minimally invasive diagnostic and prognostic markers. I hope that what I just told you helps you to understand why studying RNA, and especially also the non-coding part of the RNA, can support your research in different ways. It can help you to understand the biology behind the disease or the mechanism behind gene regulation. Gene signatures that can be used for cancer diagnosis and or prognosis can be identified. And to give an example, we have the Oncotype DX test. It is a molecular diagnostic test that analyzes the individual biology of a breast cancer tumor by examining the activity of 21 genes in the tumor tissue. And the outcome of this measurement can predict the risk of recurrence for the patient. Finally, as I stated in my previous slides, especially non-coding RNAs can be excellent minimal invasive circulating biomarkers or new therapeutic targets. In the following slides, I will discuss in more detail one class of non-coding RNAs, the microRNAs. MicroRNAs are the most extensively studied class of non-coding RNAs. Major microRNAs are very short, between 21 and 23 nucleotides, and they function as negative post-transcriptional regulators of protein-coding genes by initiating the degradation of target messenger RNA molecules or by inhibiting translation of the RNA. Due to their small size, dedicated technologies have to be used to measure the expression levels of the microRNAs. In addition to this, it is technically challenging to measure them due to a number of reasons, including the high degree of homology between family members and their low expression levels, especially in circulation. On top of that, for each canonical microRNA, sequence variants called isomers exist. The largest part of the sequence of an isomer is identical to the canonical or major microRNA sequence. However, they differ by having additional nucleotides at their 5 or 3' end or by polymorphisms in the sequence. So this is shown here in this graph. Here you can see an example, here you can see an example of a major or canonical microRNA, And here are some isomers displayed. So here you see an isomer with a different 3' end, here isomers with a different 5' end. To date, there are various platforms on the market to measure microRNA expression levels. To evaluate the performance of the available platforms, Dr. Peter Mestak, postdoctoral researcher at Ghent University and senior data analysis expert at Biogazelle, set up a study with an unprecedented scale. No less than 12 different platforms were included. This study was called the MicroRNA Quality Control Study and published last summer in Nature Methods. A follow-up on this study is currently ongoing with the evaluation of some additional platforms. As mentioned before, the primary goal of the study was to compare commercially available microRNA gene expression technologies. Included were different qPCR-based platforms, a number of hybridization-based platforms, and a limited number of sequencing technologies. In addition to this, the authors of the paper aim to develop objective and robust performance metrics, set up a framework for platform comparison including standardized samples, and support the selection of the best platform taking into account the particular study goals. Each technology provider was asked to measure the microRNA expression levels in the same set of standardized samples. Raw data were delivered and analyzed using the performance metrics that were developed. I will not go into the detailed results of the study, but if you are interested, you can check the paper that is freely available on the Biogazelle website in the Knowledge Center section. Let me just sum up the main conclusions. So each platform has its own strengths and weaknesses, and there is no single best or worst platform. The selection of the optimal platform is partly dependent on the particular application and goals of the studies. So, for instance, some platforms will be better suited to handle low input amounts of total RNA coming from body fluids. Specific platforms are more suited for wide genome profiling studies, while other platforms are better suited for targeted microRNA expression analysis. Other things you might want to consider when choosing the platform for your study are cost, throughput, ease of use, etc. Let me now introduce you to the workflows and technologies that Biogazelle applies for microRNA gene expression analysis. I have already used a number of times the words discovery and validation. If we place these two terms in a microRNA biomarker discovery workflow, then phase 1, or the discovery phase, is the search for potential biomarkers. During this phase, a genome-wide microRNA profiling is performed on a relatively small, however with sufficient power to obtain statistically significant results, cohort. The goal of this phase is to determine differentially expressed microRNAs between sample subgroups, for instance, healthy individual versus diseased patients or treated versus untreated patients. For this phase, we use small RNA sequencing. During the second phase, or the validation phase, the expression levels of the differentially expressed microRNAs determined during the discovery phase are measured on a large cohort of samples. This is a targeted approach since the microRNAs of interest are already identified. And for this approach, we use the miScript qPCR system from QIAGEN. Please note that this two-stage approach and the technologies can not only be applied for biomarker discovery studies, but also for other applications. For instance, if you want to identify microRNAs involved in embryonic development, or if you want to study a disease mechanism. Interesting to know is that microRNAs are relatively resistant to RNA degradation. This results in the successful measurement of microRNAs in various challenging sample types, including formalin-fixed paraffin-embedded tissues and body fluids such as serum, plasma, urine, saliva, sputum, cerebrospinal fluid, synovial fluid, and stool supernatants. The most important practical advice to keep in mind is that you need to use an RNA extraction kit that enables the extraction of total RNA, including the small RNA fractions. We have very good experience with the RNA extraction kits from QIAGEN. To extract RNA from FFPE tissue samples, we advise to use the miRNAs FFPE kit. When we purify RNA from body fluids, we start with only 200 microliter of the body fluids of interest, and we use the miRNAs serum plasma kit. If you measure the RNA integrity, you will see that the total RNA purified from those challenging biomaterials is fragmented. As mentioned before, this is not really a problem to measure microRNAs. However, we advise to use in the same study only samples with a similar quality. To put it in other words, it's not advised to directly compare results coming from degraded RNA samples with results coming from high-quality RNA samples. In addition to this, it might be interesting to set up a pilot study in which you include positive control samples in which you expect certain genes to be expressed, and negative control samples in which you expect certain genes not to be expressed. This will help you to evaluate the results in view of the RNA quality. I will now discuss in some more detail our whole genome microRNA profiling workflow using small RNA sequencing. Let me start with a small introduction to RNA sequencing. Dedicated workflows, especially in terms of the library prep methods, are necessary depending on the portion of the transcriptome you want to explore and depending on the sample type that needs to be analyzed. In this figure, we try to visualize which workflow we apply for which RNA molecule and on which type of sample we can offer the workflow. So, in the left part of the figure, we focus on long RNAs, including both messenger RNA and long non-coding RNAs. The most straightforward approach is the poly-A plus RNA sequencing. A secondly widely applied workflow is total RNA sequencing. Both these library prep methods are applied on high-quality RNA coming from fresh tissues or cells. For RNA coming from body fluids, for instance serum, urine, and so forth, or from formalin-fixed paraffin-embedded tissues, it is known that the RNA is degraded and fragmented, so therefore other methods are better suited for RNA sequencing of long coding or long RNAs coming from those tissues. At the right side of the figure, we focus on the small RNAs, including MicroRNAs, which are the most extensively studied, but also other small RNAs such as tRNA fragments, pRNAs, and small nuclear or nucleolar RNAs. For those types of RNAs, we have a small RNA sequencing workflow up and running for fresh, frozen, and fixed tissues, and for cells or body fluids. For our standard workflows, we use the TrueSec library preparation kits from Illumina combined with only 100 nanogram of high-quality total RNA as input material. We make use of the Illumina NextSec 500 instrument. For the data processing and analysis of our RNA-seg data, we have our own proprietary pipeline, Cobra. This pipeline is built up out of different modules, allowing data processing of different applications, including small RNA sequencing. Zooming in a little bit more on our small RNA sequencing workflow, this workflow is optimized to identify not only microRNAs, but also other classes of small RNAs, as shown in this frequency histogram that shows the number of reads in function of the RNA fragment length. the microRNA fraction is represented in green as the smallest molecules ranging from 20 nucleotides to 23 nucleotides. And in addition, we have a second peak ranging from 25 to 36 nucleotides and including mainly tRNA fragments in pink, but also pVRNAs in light blue and small nuclear and nucleolar RNA fragments in yellow. In the following slides, I will show some RNA-seq results we obtained on challenging samples in our ongoing R&D efforts. As I mentioned earlier, our standard workflow starts with 100 nanogram of total RNA. However, in some cases, 100 nanogram of total RNA is not available. And therefore, we have tested and optimized our small RNA sequencing workflow on ultra-low input amounts, which is 10 nanogram of total RNA, equivalent with the RNA amount obtained from 500 cells, and on 1 nanogram of total RNA, equivalent with the RNA content of about 50 cells. Two criteria that we have evaluated are reproducibility and sensitivity. In terms of reproducibility, comparing the normalized read counts we obtained per gene for two independent library preps for the same sample, we obtained a very good reproducibility as shown in this graph for 10 nanograms. In terms of sensitivity, defined as the number of microRNAs that are detected, starting from 10 nanograms, 75% of the microRNAs that we normally detect in using 100 nanograms of that same sample were still quantitatively detected starting from only 10 nanograms of total RNA. In addition, we have also applied our small RNA sequencing workflow on RNA derived from serum and other body fluids. We start with isolating RNA from only 200 microliter of serum and use half of the extracted RNA in our library prep. And following this approach, we detect between 500 and 800 microRNAs depending on the read and the nature of the sample. To give an example on a small set of samples, so when 15 million reads per sample were generated, on average around 500 microRNAs were detected, while when 25 million reads per sample were generated, we were able to detect on average 800 microRNAs per sample. Evaluation of the microRNA correlation between sample replicates again shows a high reproducibility over a wide dynamic range, a dynamic range of about 15 log2 units. Although we were already quite satisfied with those results, we further optimized our workflow in an attempt to increase our efficiency for the specific detection of microRNAs with a reduction of the sequencing cost. And we have now reached a protocol in which we generate 30% more microRNA reads compared to our standard protocol on body fluids. And this resulted in 15% more microRNAs that were detected, as shown here in green the standard protocol and in pink the optimized protocol. Let me now discuss a little bit more our data analysis workflow for small RNA sequencing. So we distinguish in general three levels of transcriptome data analysis: primary data processing, basic data analysis, and advanced data analysis. Primary data processing involves the conversion of raw data into more meaningful normalized expression levels. And although it does typically not yield the final results you are interested in, it is an essential step and care must be taken to ensure that proper data is generated for downstream interpretation. For small RNA-seq, FASTQ files are converted into counts or FPKM values using Cobra. The next level is basic data analysis. It takes the process data from the previous level for differential gene expression and statistical analysis. Cobra can be used for standard analysis, but for more customized or advanced statistics, our data analysis experts rely on the statistical programming environment R. Basic data analysis is sometimes enough to answer many research questions. However, with our advanced data analysis, we can, however, generate more value from your data. For example, linking experimental data to pathways and gene functions yields more insights. Another example of advanced data analysis is the identification of diagnostic or prognostic gene expression signatures. So Cobra is a software suite that we developed to manage, process, and analyze RNA-seg data. And we believe quality control is an essential part of transcriptome analysis. For small RNA sequencing, Cobra summarizes the quality controls in 3 graphs. So the first quality control step inspects the technical quality of the data. What fraction of the reads can be used for downstream analysis? So here the green bars are the reads that pass QC. A second graph of quality control report is on the outcome of the mapping step. So what fraction of the reads that passed QC map to microRNAs or other small RNAs? So here shown in green for the microRNAs, in pink for the other small RNAs, and which fraction cannot be annotated or even not mapped. Both technical and biological variables contribute to the distribution of reads in these 4 classes. And the third graph gives a more detailed view of the distribution of the mapped reads for a single sample, as I discussed before. Cobra does not only generate count tables for canonical microRNA sequences, but also reports on isomers and other small RNAs. I have included the sequencing results to highlight the importance of looking at the isomer details as well. So in this example, we see that only a fraction of the reads map to the canonical microRNA in pink, whereas the 3 isomers on top are responsible for the bulk expression of this microRNA. And since different isomers may have different functions or effects, one should also look at them. I would like to end this part of the presentation with an example of a study we recently have performed in a collaborative research project. So the study was set up to discover non-invasive biomarkers that can be used to diagnose the inflammation status in patients with a certain inflammatory disease. To this goal, microRNA expression levels were measured in serum samples coming from 83 patients diagnosed with the inflammatory disease and for which the inflammation status was clinically determined. The discovery phase of the study included 83 patients, of which 14 had an acute inflammation, 17 a chronic inflammation, and 51 patients did not have inflammation. MicroRNAs that are differentially expressed in function of the inflammation status were determined. The figure shows the number of differentially expressed microRNA in function of the inflammation status. So, to give an example, between patients with a chronic inflammation status and normal controls, 22 microRNAs were identified to be differentially expressed between those subgroups. Between patients with an acute inflammation status and patients with a chronic inflammation status, 3 microRNAs were detected that were differentially expressed. Based on the differentially expressed microRNAs between the 3 patient subgroups, 3 inflammation signatures were constructed. One that discriminates between patients with acute versus chronic inflammation, one that discriminates between patients with acute inflammation versus normal, and one that discriminates between patients with a chronic inflammation versus patients without inflammation. As can be appreciated from these box plots displayed in the slide, each of the three inflammation signatures shows a significantly different signature score between the different patients groups. From this data, we conclude that small RNA sequencing on body fluids is an interesting strategy to identify non-invasive biomarkers for disease. The obtained signatures need to be further validated on an independent cohort in a targeted approach. And this brings me to the next topic of this presentation. I will further discuss now the technology and the workflow that we apply for targeted micro-expression analysis in validation studies. The platform we use is the miScript PCR system from QIAGEN. This is a qPCR-based approach to measure the expression levels of specific microRNAs of interest. Besides individual assays, QIAGEN also offers arrays for biologically relevant pathways and whole miRNA panels. In total, 2,405 validated human microRNA assays, which is for 93% of the current miR-based content, are available. The workflow starts with a universal reverse transcription step, which means that you only need to perform one single RT reaction to study the whole miRNA. The system shows a high sensitivity to detect microRNAs. And in addition to this, an optional cDNA pre-amplification step can be included. This is definitely of interest when studying microRNA expressions in body fluids or when low RNA sample input amounts are available. Inherent to the workflow are a number of synthetic spike-in microRNAs which allow quality control during each step. And I will come back to this later in my presentation. Just a brief overview of the workflow. So the workflow starts with a universal reverse transcription reaction to convert the microRNAs to cDNA, followed by a pre-amplification of the cDNA. And as mentioned, this is optional when working with small input amounts. Subsequently, the amplified cDNA is used as a template for the qPCR reaction using the miScript PCR assay. After running the qPCR reactions, the raw data under the form of CQ values should be converted in normalized data to allow interpretation of the results. During every step of the microRNA profiling workflow, synthetic microRNA spike-ins are included to allow quality control. During the RNA extraction, a synthetic C. elegans microRNA can be included to assess the RNA extraction efficiency. In the master mix used for the reverse transcription, a microRNA reverse transcription control is included to assess the RT efficiency and the sample purity. qPCR inhibition, technical reproducibility, and interplate variation are evaluated based on a positive PCR control assay, the PPC assay, included in the qPCR master mix. Once the raw data or CQ values are obtained, proper data normalization has to be performed, and this step has the largest impact on the final accuracy of the results. We use the multiple reference gene normalization strategy for data normalization, and this method is described in the Genome paper published in Genome Biology in 2002 and currently cited more than 8,000 times. This method identifies the most stably expressed genes and then uses the geometric mean of these microRNAs to normalize data. In sake of time, I will not go into more detail around this topic, but I can refer to a blog written by Professor Jo van de Sompelen and published on our website. Both the selection of stably expressed microRNAs and the data normalization can be performed using our in-house developed qPCR data analysis software Cubase Plus. Cubase Plus is a user-friendly software package that helps you to analyze your qPCR data. All you need to do is upload the raw data or the CQ values and coming from the qPCR instrument and follow the experiment wizard. Again, if you want more details about the software tool, I can refer to the Biogazelle website, where you can find a nice webinar video explaining the software possibilities. I will end this part of the webinar with 2 customer studies performed at Biogazelle using the miScript PCR system. The goal of the first study was to set up a novel molecular taxonomy for colorectal cancer based on microRNA expression patterns. To this end, more than 650 formalin-fixed paraffin-embedded colon tumor biopsies were profiled. The study started about 3 years ago. Back then, the most comprehensive panel of microRNAs targets for screening purpose was the LifeSeg panel consisting out of 75 microRNAs. Afterwards, the panel was extended to obtain data for more microRNAs. Additional microRNAs were selected based on literature and TCGA public data. The expression levels of these additional microRNAs were measured using the miScript PCR system. Customized pre-spotted arrays were used containing miScript assays for 180 microRNAs of choice and a number of control targets. In the next slide, I will show some quality control metrics that we applied on the data. So the miRTC assay that is used to assess reverse transcription performance was measured in duplo for each sample. Aberrant miRTC signal is suggestive for sample-mediated RT inhibition. And so the average miRTC CQ value across all samples for 2 batches of samples is shown on this slide. In both batches, one sample shows an aberrant miRTC CQ value indicating PCR inhibitors present in these samples. To assess PCR performance, the synthetic PCR template present in the qPCR mix was measured with the PPC assay, in duplo again for each sample, and the average PPC CQ value points to a uniform detection across the array. To further evaluate data quality, we calculated the number of detected microRNAs, and in both batches of samples, one sample with an abnormal low number of detected microRNAs was detected. Finally, the mean microRNA expression per sample was calculated, again for one sample in each of these batches. A lower mean microRNA expression, resulting in a higher CQ value, was observed in line with the results obtained with the miRTC assay and the number of detected microRNAs. So in conclusion, using the miScript system, we have numerous quality controls in place to detect and exclude spurious results. In terms of results in this study, our collaborators could recognize 4 molecular subgroups of colon cancer patients. On the left, you can see a clustered heat map with red pointing at high expression and blue at low expression. The 660 samples are in the rows, and the microRNAs are in the columns. On the right, you see the results of group identification algorithm indicating to optimal separation is achieved with 4 subgroups. Interestingly, the subgroups are defined by microRNA expression levels are different from the subgroups previously defined by mRNA expression. So this points at independent information encoded in the miRNA. Another example of a study in which we used the miScript PCR system was set up to identify microRNAs that can predict relapse in a certain inflammatory disease. To this end, a pilot study was set up, including 12 patients with stable remission and 12 patients with unstable remission, 366 selected microRNAs were measured in two sample types being serum and stool supernatant to determine which sample types give the best chance to identify predictive microRNAs. From the 366 measured microRNAs, on average 118 or 31% of the microRNAs are detected per sample. Overall, 159 microRNAs are detected in at least one serum sample, while 365 unique microRNAs are detected in at least one stool sample. Differential microRNA expression analysis results in a number of differentially expressed microRNAs between stable remission and unstable remission. This can be seen in the volcano plots, showing minus log 10 p-values in function of the log 2 fold change. Green points indicate significantly differentially expressed microRNAs in serum on here and in stool in this part of the figure. In conclusion for this pilot study, a similar number of differentially expressed genes in serum and stool could be identified, and so we decided to do the full study on both sample types. This will be the first study of its kind in analyzing microRNAs in both serum and stool on such a large scale. So, this brings me to the last topic of this webinar. So during these last minutes, I would like to summarize how Biogazelle can support your R&D projects on each and every level. We aim to build up long-term research collaborations by offering excellent project management and communications while performing a project. Collaborations can start with proof-of-concept studies that are the basics to larger-scale projects and or recurrent projects. Biogazelle offers total solutions for the study of RNA expression levels, starting from RNA extraction over measurement using state-of-the-art and validated technologies to understandable results. Depending on the type of study or the recent question to be answered, we help our customers in choosing the best platform. We stand for quality. All the platforms and workflows that we offer are fully in-house validated, and our lab works according to the ISO 17025 requirements. Moreover, both the whole genome microRNA profiling workflow and the workflow for targeted microRNA expression analysis is ISO 17025 accredited. In addition to this, we have an extensive track record in microRNA biomarker discovery. Our expertise is based on the successful profiling of thousands of samples, including a large number of body fluids. We are partnered for microRNA profiling in multiple European research consortia, and we publish numerous peer-reviewed publications. On top of that, we can show a wide range of references in academic top research centers and in the biotech pharma market. Let me conclude with 5 good reasons for choosing Biogazelle as your research partner. We want to think together with you. We have the latest technologies in-house. We are used to work with body fluids. We have the right setup to support clinical trials, and we understand the challenges in biomarker discovery as we have our own R&D and discovery projects. With this, I will end my presentation. Thank you all for joining us today, and I think we still have some time to take some questions.
Okay, thank you, Ariane, for a very interesting and enjoyable presentation. So, first, let's take a quick look at the poll results before we move on to the Q&A session. So, first, let's have a look at the results of the question. Where do you typically send your microRNA profiling work? So this shows us that 50% of our listeners perform microRNA profiling in their own lab. Ariane, are you surprised by this response?
I think this depends on the type of researchers. I believe that academic researchers mainly do this in-house. So maybe a large part of the audience today is academic.
Cool. Now, moving on to the results of the question, where in the research process are your current project or projects? And we can see that most people are working on biomarker discovery, and second to this is functional validation of microRNA. Do you have any comments on these results, Ariane?
Yeah, I think for the people that are involved in biomarker discovery, it's definitely interesting to use a wide screen technology like small RNA sequencing. It will give you interesting results. It's an unbiased technology to identify all the microRNAs present in your sample.
Great, thank you. And now let's look at the results of the final question. What product strength is most important in your microRNA studies? And we can see that over 50% have answered specificity. Is this what you would expect, Ariane?
Yeah, I think this is very important, that you really can identify the microRNAs, the specific microRNAs. Yes, I'm not surprised by this answer.
Great. Now let's move on to the final part of today's webinar, the question and answer session. Please submit any further questions by clicking on the Ask a Question button at the bottom of your screen. Okay, Ariane, the first question we have is, when performing studies using a normal comparative cohort, what is the cohort size and how is it defined, i.e. clinical criteria?
This really depends. This is not a real straightforward answer. The larger you take the cohort, the better chances to have to obtain statistically significant results. If we need to put something on it, then I would say at least 20 controls versus 20 patients is the minimal you should start with. The best approach to define this is to perform a power analysis, but if you cannot do that first, then I would say start at least with 20 versus 20.
Great, thank you, Ariane. Next question. Do microRNA signatures that discriminate inflammation track with mRNA signatures in the discrimination of the same patient cohorts?
Can you repeat this question?
Yes. Do microRNA signatures that discriminate inflammation track with the mRNA signatures in the discrimination of the same patient cohorts?
In that study, so that was the first case study that we have discussed, we have not yet done messenger RNA sequencing on the serum samples. So I cannot really answer on that question.
Okay, thanks, Ariane. The next question. How well can NGS technology for microRNA be trusted for microRNA quantification analysis, or do you always recommend quantifying using qPCR?
It depends. We believe in the strength of the technology, so it's not necessary to do a technical validation. However, as I emphasized different times during this webinar, it's necessary to do a biological validation. So meaning that you need to validate your results on an other cohort, an independent cohort. And for that, we always use qPCR.
Great, thank you. Next question. Can we use this system for short hairpin RNA validation and assessment in the cell and tissue also? So it's a bit a double question. I'm not sure what is meant, but if you want to check whether if the short hairpin is used to knock down a certain microRNA, and you want to check the expression of that microRNA, you can sure use the miScript technology. On the other hand, if you want to check the expression of your short hairpin RNA, then you would need customized assays. Thank you. Can you talk about the benefits of microRNA profiling?
Yes, I see benefits for both studying microRNAs as well as doing a profiling study. So microRNAs have been shown to be very important negative regulators of gene expression and do this in a time and place specific manner. They are crucial in normal development and disease processes. So starting a profiling study is typically done when it's not known in advance which and to what extent microRNAs are involved in your experimental conditions. So important for a profiling study is to measure as many microRNAs and possibly isoforms as possible in order to maximize the chance to finding interesting results.
Thanks, Ariane. We also have someone thanking you for the useful presentation, and they have a question. Can we use this for other RNAi research validation?
Can we use what? Or what is
I guess they just say, can we use this for other RNAi research validation?
So, yes, for RNAi, so RNA interference validation, if you want to check whether your RNA interference works, yes, you can use the miScript system.
Great. We can also follow that up via email as well, if that's useful. Next question. If I want to do an entire miRNome, how much sample would I need to begin? What are your thoughts on using pre-AMP, and does this affect the reliability of data?
So what was the first part of the question was how much sample you need for an entire miRNA, correct?
Yes, that's correct. How much sample?
So we typically start our workflows with 100 nanogram of total RNA. So this is equivalent with about RNA coming from 2,000 to 5,000 cells. A second part of the question was about the preamplification, I believe.
Yes, that's correct.
We normally use preamplification when substantially lower, when we have low input amounts of material. And we have extensively compared preamp versus no preamp and have seen very good concordance in the measured fold changes among samples. So when you include the preamplification step, you really don't need a lot of RNA. I would say 10 nanogram of total RNA from tissues and only like 10% of the RNA extracted from 200 microliter of body fluid.
Great, thank you, Ariane. The next question. Can you discuss the normalization step for microRNA expression study in body fluids and suggest a best method?
Yes, for body fluid, it's about normalization in the discovery. Then you can apply the same kind of normalization in small RNA sequencing. Also, if you use qPCR, miRNAs, you could apply global mean normalization, and that would be the best normalization strategy in body fluids, the global mean normalization.
Great, thank you. Next question, someone asking, can I use data from NanoString to find reference genes using QBase?
No, you cannot import your NanoString data in QBase Plus. But however, you can use your results, your NanoString results, to select stable reference genes, but not using QBase Plus.
Okay, great. Thanks, Ariane. Next question. When you chose to use the QIAGEN microRNA platform, can you talk about why you made that decision?
I think I elaborated a little bit on this in the presentation. There are a number of interesting characteristics of this QIAGEN platform. First of all, the universal reverse transcription reaction. Definitely also the fact that they have assays for almost the entire miRNA. And on top of that these assays are wet lab tested. Also interesting for us was the preamplification step, because we have a lot of projects with low input amounts of total RNA. And on top of that, also there was a very good overall good performance according to the MicroRNA study and internal evaluations.
Thanks. We have time for one more question. And the question is, can I design custom assays after sequencing if I find novel microRNA species?
Yeah, that's actually a question that QIAGEN would probably can better answer, but yes, I believe they have the ability to design custom assays for further quantification of novel microRNAs. They have a custom design tool, yes.
Great, thanks Ariane. Well, that's all we have time for today, and thank you very much Ariane again for today's informative discussion and presentation. Thank you to everyone joining us online and for your interesting questions. I hope that you found this session worthwhile. If we didn't manage to answer your questions, feel free to email me at sarah@selectscience.net and I will follow up with your question personally. If you'd like to listen to today's webinar again or to invite a friend to listen, it will be available to watch on demand shortly. Goodbye and thank you once again for joining us.